All right. Well, as I mentioned in class today, I think you're really gonna like the information that we're gonna talk about next week, beginning here. And so this first chapter in the book covers the cell cycle, and inside of the chapter they do a quick review, a crash course, if you will, on mitosis. If you don't feel comfortable about the 5 stages of mitosis, I encourage you to go over that. It's about a page of that chapter. I won't talk about it at all in this video. Similar to what I did for today in class, this video will provide us a framework, a foundation, and we'll dig a little bit deeper on Monday and talk about some of these topics in more detail. All right, so key terms. First key term we need to talk about is M phase. M phase, also known as mitosis plus cytokinesis. So within the cell cycle, you have interphase, which is composed of G1, S, and G2. Although for most cells in our body, we'll talk about this in a minute, it's actually G0 or G naught. And then M phase would be mitosis plus cytokinesis. You have a set of enzymes called cyclin-dependent kinases, and kinases are enzymes that phosphorylate other proteins. And cyclin-dependent kinases, these are kinases that are only active in the presence of a particular type of cyclin, which is another protein. And these collectively, the genes that code for these proteins are collectively referred to as proto-oncogenes, which is basically the preliminary cancer-causing genes. And so in cancerous cells, a lot of these cyclin-dependent kinases are overexpressed, are overactivated, and just continue to keep that cell moving through the cell cycle stage after stage after stage, facilitating that rapid, uncontrolled growth that you find in cancer. You have a group of proteins called cyclin-dependent kinase inhibitors that function to ensure either that the cyclin that cyclin-dependent kinase binds is not active or present, or that they may phosphorylate or remove phosphate groups from the kinase themselves and therefore inhibit those kinases. These are collectively, or at least the genes that code for these are collectively referred to as tumor suppressor genes, because when they are being expressed, these cyclin-dependent kinases are not active and the cell is not moving through the cell cycle. Phosphatases, these are enzymes that remove phosphate groups from other proteins. Many of the cyclin-dependent kinase inhibitors are phosphatases, and many of them are inhibited by phosphatases. And so you get in this just a snapshot of what you oftentimes have in enzymes that have an on-off switch. Oftentimes that on-off switch is the presence or absence of a phosphate group. So some of these enzymes, they are active when they are phosphorylated. Some of these enzymes are active when they are dephosphorylated. And so it's a common theme to see these phosphate groups being moved around to function as on-off switches, which, if you remember what's constantly happening inside cells, and that is hydrolyzing ATP, you get the sense that there's a lot of phosphate moving around and changing from one protein to another. And so it's actually a great way to facilitate an on-off switch for these enzymes. Checkpoints. Within the cell cycle, there are 3 main checkpoints, and they all require a very specific cyclin-dependent kinase and cyclin complex. One of them, you'll see we refer to it as a different name. The other 2 checkpoints, we'll talk about a very specific, we'll call it a CDK, is what is short for cyclin-dependent kinase. You'll see a CDK and then a cyclin combo, but this one one of them we actually refer to as something else, and that's this one. And this is M-phase or mitosis promoting factor. And this is a cyclin-dependent kinase, a CDK, bonded with a specific cyclin. But because it does so much for triggering M-phase, we actually refer to it as M-phase promoting factor, give it a special name. MAPS, these are microtubule associated proteins, and these are proteins that function to alter the way microtubules are assembled. And we'll see this in a couple of slides, but something you need to know about microtubules is in a normal cell that is not going through M phase, it's in G1 or it's in G0, the microtubules are very stable structures. You'll remember from cell biology that the microtubules provide the scaffolding for motor proteins to walk across, and so they remain very stable. During M phase, they are incredibly unstable. And so part of that is actually facilitated by triggering a different set of microtubule associated proteins. We have what's called the pericentriolar matrix. If you'll remember the centrioles that migrate to opposite poles of the cell, where the mitotic spindle radiate out from during M phase, these centrioles act as what's called a microtubule MTOC, so it's microtubule origination center, and it's the primary one in animal cells that are these centrioles combined together to form the centrosome, and around the two centrioles, and we'll see an image of this in a few slides, there's a matrix full of a number of different types of proteins that facilitate building microtubules out from that, the centrosome, from that microtubule organization center or originating center. APC is Anaphase promoting complex, and this is what allows the cell to move from metaphase into anaphase. And this is where you start to see the degradation. When you get the anaphase promoting complex going, you start to see the degradation of the M phase promoting factor, and you start to see the cell moving towards the end of M phase. All right, so here's an image of a cell cycle, and you can see the three main checkpoints. Now, there are more than just these three checkpoints, but these are the three main checkpoints. And so let me get my marker here, and I will make sure it is not red. We'll make it a nice rich blue again. Here's G1, and G1 is a checkpoint that allows the cell to either move from G1 to S or from G0 to S, seeing as most cells are in G0, that they're basically have left the cell cycle until some kind of a growth factor or some really unique physical contact that cell receives triggers it to move from G0 into S. So the cell can move from G0 into S or from G1 into S, and in order to do that, it has to go past this G1 checkpoint. And that G1 checkpoint checks for fidelity in the DNA strands, and in order to move past that, you have to have a very specific CDK, cyclin-dependent kinase, and cyclin complex. During S phase, the DNA is duplicated, the centrioles are duplicated, and then you move into G2. In G2, the centrioles are elongated, they are basically matured to ready the cell for M phase, and then you have G2 checkpoint, which now it doesn't just check for errors in the DNA, but it also checks to ensure that the two copies of the DNA are identical. And in order to move past G2, you need a very specific CDK cyclin complex, and then you can move into M phase, which again is a combination of both mitosis, which is karyokinesis, the separation of the nuclei into two nuclei, or the separation of the nucleus into two nuclei, and cytokinesis, the separation of the cell into two cells. And here's the third checkpoint, the mitotic checkpoint, or the M checkpoint. And there the cell checks to ensure that the two sister chromatids on each chromosome are attached to opposite poles. And we'll see that in more detail in just a minute. Okay, so this at first glance seems very, very complicated, but I want to just walk you through this. So here is M phase in the cell cycle, ushering over to G1, and then you have this G1 checkpoint that in order to go from G1 to S, you need a very specific cyclin-dependent kinase cyclin combo, and that is right here, CDK4 and CDK6, so cyclin-dependent kinase 4, cyclin-dependent kinase 6, combined with cyclin D. And so once you build that complex, then the cell can actually move from G1 into S phase. Same thing is true for a cell that's in G0, but in order for a G0 cell to actually start to transcribe the genes that code for CDK4 and CDK6 and these various cyclins, you have to have something very specific happen. You have to have growth factors or the cell combine becoming anchored to another cell that triggers this, and then it will express this cyclin D, which can bind with cyclin-dependent kinase 4 and 6. It'll also trigger the downregulation, that's what this arrow is here. It'll trigger the downregulation of these cyclin-dependent kinase inhibitors. That's what CKI stands for. And what you'll find is these oftentimes, these proteins that act as cyclin-dependent kinase inhibitors are just named P and some number. So in here we've got P15, P16, P18, and P19 that are all cyclin-dependent kinase inhibitors. And yeah, these growth factors can trigger a cell that's in G0 to downregulate the expression of these cyclin-dependent kinase inhibitors. So that will activate CDK4 and CDK6, and then at the same time the cell is expressing cyclin D that can then combine with this to trigger the several other processes. And so this complex here triggers or activates this gene called PRB, which activates E2F, which are initiation factors that lead to the transcription of several factors. And ultimately, the thing you have to know is this one right here. So it leads to the transcription of a number of different factors, but the big one here is it leads to the transcription of cyclin A that combined with cyclin-dependent kinase 2 to actually trigger the duplication of DNA. So notice where this arrow connects right here is actually into S phase, where that process has already started. And so this complex here, this cyclin-dependent kinase 2, along with cyclin A, ensures that the DNA is duplicated, that the DNA is duplicated correctly, and that the DNA is duplicated only once. And so that doesn't happen unless you build this complex here. This complex doesn't form unless you trigger the production of E2F, and you can't trigger the production of E2F without activating cyclin-dependent kinase 4 and 6 and combining that with cyclin D. Now notice there's another thing, so this you don't have to know, but I just want to point this out, that this initiation factor also triggers the production of cyclin E, and that cyclin E acts as a positive feedback mechanism by combining with cyclin-dependent kinase 2 to actually lead to the production of more E2F. So it acts as what's called a positive feedback loop to continue to make more E2F, which will make more cyclin A, which will combine with more activated CDK2, which will trigger the production of a second copy of the DNA. All right, so again, just to sum all this up, a cell, in order to go from G1 to S phase or to go from G0 to S phase, you have to form this complex here, the CDK4 and CDK6 combined with cyclin D. Now, one other thing I want to point out here, if there's DNA damage or if the cell is exposed to low oxygen conditions, it will produce this tumor suppressor gene, or the product of this tumor suppression gene called P53, and that P53, that's up arrow, as it's being upregulated, it will trigger the production of several other cyclin dependent kinase inhibitors, and they will shut this down. Even if the cell currently is making this, it'll shut that process down. And so that's where you get that G1 checkpoint. If the DNA is damaged, the cell will make P53 and it will shut this process down. Now, what you find is in almost all cancers, there's a mutation in this P53 gene. And so the cell loses the ability to check it can still check for DNA damage, but it loses the ability to shut down the cell cycle in response to finding damaged DNA. All right, so here's the M phase promoting factor, and again, this is another. It's CDC2, which is also known as CDK1. It's just two analogs in two different organisms. So this CDC2 is you can think of it. It's synonymous with CDK1. This right here is cyclin B, and so when you get CDK1 combining with cyclin B, you form an inactive form of the M phase promoting factor. Now, this M phase promoting factor is phosphorylated by two different kinases, one that is an activating kinase, one that is an inhibitory kinase. So then you get this kind of really interesting state here where the M phase promoting factor is both inhibited and activated at the same time. And so in order to actually allow it to be active, you have to use a phosphatase to remove that phosphate group. And here's this phosphatase here, CDC25, and CDC25 will remove that phosphate group, and now you have an active M phase promoting factor. Now, here are some of the things that this does, and again, I told you at the beginning of the video, the reason why we give this a special name rather than just saying CDK1 cyclin B, that complex, is because it activates so many things and it triggers all of what happens during M phase. So here, one thing is it activates the gene that codes for CDC25, so it actually acts as a positive feedback, making more molecules of M phase promoting factor. It phosphorylates lamin A, B, and C, and if you don't remember these are proteins found on the inner nuclear membrane, and they stabilize the nuclear envelope. So when you phosphorylate them, it leads to the degradation of the nuclear envelope. Lamin A and C end up suspended, freely dissolved in the cytosol. Lamin B remains inside of pieces of the nuclear membrane, now separated into thousands of vesicles. It also inhibits an enzyme called actomyosin ATPase, and this enzyme functions to keep the cytoplasm near the plasma membrane moving and alternating between a fluid state and a solid state, which keeps the cell relatively oval. But when you inhibit this, it rounds the cell out a great deal, which actually allows for very efficient production of the mitotic spindle. It also phosphorylates condensins and histone complexes, and this is going to function to condense the DNA fully. And so we talked about this on Monday, and so you can go back and look at what fully condensed DNA looks like. And this is all triggered by M-phase promoting factor. It also phosphorylates the microtubule associated proteins, and these create instability in the normal cytoskeleton. Those microtubules fall apart, the tubulin proteins are released, and then you can start building the mitotic spindle, rebuilding microtubules, but only radiating out from the centrosomes that have migrated to opposite poles of the cell. All right, so here is a centrosome. Here are the two centrioles here, and the two centrioles are physically attached together by some protein fibers. Here is the pericentriolar space, this gray area around these. And here is where you have the mitotic spindle form. So these microtubules grow out from here. There are several proteins in this pericentriolar matrix that facilitate attaching those two different types of tubulin together to form microtubules. And as we'll see in maybe the next slide, we really get three different types of microtubules that radiate out from these. Now, remember we duplicate the centrioles during S phase as well, and then so they migrate to opposite poles, and so on both sides of the cell you have microtubules growing out and radiating from those centers. All right, so here's referencing the centrioles. Here's a cell in G1 or G dot where you have got the normal condition of a single centrosome made up of those two centrioles. And again, even in G naught and G1, those are the main microtubule organization center and microtubule origination center. During S phase, the centriole or the centrosome is duplicated. Over in G2, these are elongated and matured to ready them to make the mitotic spindle. And then during M phase, they are separated and migrate to opposite ends of the cell. And that's what's illustrated here. And so now let's go look at the three types of microtubules. So again, one of the things that M-phase promoting factor triggers or activates are these microtubule associated proteins. And these function to build these three types of microtubules. So you have astral microtubules, and this is what I mentioned today in class, are the microtubules that we still have really no idea what they do. And then you've got what are called kinetochore microtubules, and these are attached to the kinetochore, which is a multi-layered protein scaffolding that attaches to the syn proteins around the centromeres. And so you get these will grow out, these kinetochore microtubules will continue to grow out away from the centrosome until they make contact with the kinetochore. At that point, they will stabilize. They will not lengthen anymore, although they may still move that chromosome around during this process. You also have what are called polar microtubules, and these are not physically attached to a chromosome. Instead, they are actually physically attached via a motor protein to a polar microtubule radiating out from the other centrosome. And so you'll see these will function to elongate the cell during anaphase. And so again, once these make physical contact via this motor protein with another polar microtubule, they will also stabilize until they start to function during anaphase. All right, so something very important has to happen for us to finish M phase. We have to trigger what's called the anaphase promoting complex. And so this is activated during metaphase, and it does several things. First, it just in general, this complex acts as what's called a ubiquitin ligase. And so ubiquitin is a protein that when other proteins are labeled with it, it marks them for destruction. It basically triggers them to be broken down by proteolases. And so a ligase attaches things together, creates bonds. And so the anaphase promoting complex attaches ubiquitin to various proteins to mark them for destruction. One of those proteins it marks for destruction. Wow, now my blue line is in an awkward spot. One of those proteins it marks for destruction is securin. Securin is an inhibitor of a protein called separase, and so once securin is marked for destruction, it disappears, and now since it's been inhibiting separase, when it's gone, separase can now act. Separase functions to degrade the proteins that are holding the sister chromatids together. And so when the separase can break down those proteins, the sister chromatids lose their physical connection and then can be pulled to opposite ends of the cell as two things happen, which we'll talk about in just a moment. It also marks B-cyclin for destruction. And remember, B-cyclin is the cyclin that activates CDK1 in order to form the M-phase promoting factor. So when it marks that for destruction, it inactivates the M-phase promoting factor and basically ushers the end of M-phase. So the nuclear membrane starts to reassemble. The microtubule organization centers around the centrosomes start to deteriorate. The chromosomes begin to decondense. All of these things that are triggered by M-phase promoting factors start to go back to the way they function in a normal cell. Now, ultimately, the anaphase promoting complex triggers both anaphase A and anaphase B. There are actually two different phases of anaphase. In anaphase A, the kinetochore microtubules shorten, and they actually shorten at both ends. They shorten at the end that's attached to the chromosome, and they shorten at the negative end that's attached to the pericentriolar space or the pericentriolar matrix. And so this will pull the sister chromatids to opposite poles. And then anaphase B, the polar microtubules lengthen to actually push the cell, elongate the cell, further separating these sister chromatids apart from one another. And again, you have to remember at the same time, the M-phase promoting factor is inactivated, and you're starting to rebuild the nucleus at both poles, and you're starting to fully deteriorate these microtubule organization centers, the mitotic spindle. You're starting to decondense these chromosomes, all as the chromatids are being pulled to opposite poles of the cell. All right, so here's a graph to show this. Here's anaphase A, where again these kinetochore microtubules are pulling the sister chromatids in opposite directions. And you can see that here. They're doing that as those microtubules physically shorten. And they shorten at both ends. They shorten at this end here that's attached to the chromosome, and they shorten at the negative end that is attached to the pericentriolar matrix. In anaphase B, these polar microtubules lengthen, forcing the two poles of the cell further apart. And so that functions to separate these even more. And so then as the nucleus starts to reform around these chromosomes, you have two completely separate nuclei. Karyokinesis is done, and this is telophase, and then cytokinesis can progress to separate that cell into two cells. Last thing I want to talk about is that last checkpoint. I mentioned the M checkpoint or the mitotic checkpoint, and what it is, but I want to show you it. So basically you've got this spindle checkpoint, and if it's active, it will prevent the cell from progressing through M phase. It'll keep the cell from finishing M phase. When it's inactive, it will allow anaphase to happen, and M phase will reach its completion. And it's inactive only when you have a microtubule, a kinetochore microtubule, from each pole physically attached to the sister chromatids. So even over here, you have this one. This pole is actually attached to both sister chromatids, but as long as you have this one attached here, you'll still pull this chromatid apart because this microtubule is shorter, and we'll pull it before this one has a chance to pull it towards that pole. And so that's the final checkpoint in the cell cycle, and can only be bypassed if the chromosomes are aligned correctly and are attached properly to those kinetochore microtubules. So as you prepare for class on Monday, make sure to take the quiz and be ready to actually dig a little bit deeper into these processes.